Today we're going to continue talking about antibody structure as well as thinking about antibody function. So when we left off last time, I told you about this Y-shaped protein that scientists had discovered that was sort of able to lead to protection in certain instances. That was called the antibody. And I told you some experiments last time where Bombering and Kitasato were transferring different either solutions or cells to try to find out what the protective thing was. And people after them did things like use proteases or lipases or things like that to say it was a protein. In times after that, people did additional kinds of biochemistry experiments to try to understand some parts of the structure of that protein. And so the next sort of thing that I want to tell you about are three different processes or three different things that scientists did to try to break this protein to help them understand its parts. And so we've got three different degradations that were done that really helped us understand the structure of the antibody. One of them was that they treated it with a compound called mercaptoethanol. As you can see here, it's listed as mercaptoethanol reduction. But the thing that you can know about mercaptoethanol, if you don't want to think about the word reduction, that chemistry word scares you. The point of beta mercaptoethanol or mercaptoethanol is that it breaks disulfide bonds. It breaks disulfide bonds. And so they treated antibodies with mercaptoethanol to break disulfide bonds. And when they did that, this was, the antibody that they started with, and they found out that they actually got 4 protein chains. So this thing that looked like a Y was actually 4 separate protein chains that had been held together by disulfide bonds. Those 4 chains were basically 2 chains that were identical and that were big, and 2 that were identical and that were small. And we called the 2 that were identical big the heavy chain, and we called the 2 small ones the light chain. And so this Y-shaped protein is actually a complex of 4 different proteins, 2 identical heavy chains, 2 identical light chains. And you can see the disulfide bonds holding them together. So you can see the heavy chains are in dark blue, and they kind of make this main part of the Y. They're held together by a couple disulfide bonds right there. And then we've got a light chain on either side, held together by a disulfide bond. And so this is what it looks like. In my antibody here, the heavy chains are in yellow and the light chains are in orange. And so this is sort of the overall structure they found by doing the beta mercaptoethanol reduction. They then wanted to use some other types of things to kind of learn about the structure of the antibody. And so now they use a protease to degrade the protein and to break it into different parts. The protease they use in this example is called papain. And when they did their papain digestion, they found that they got 3 pieces. One piece that was sort of the tail that you can see here, and these other 2 identical arm parts. And when they looked really closely, they found that the arm parts, which you can see are made up of a heavy chain and a light chain each, could bind to antigen. And so they called those parts Fabs, or fragment antigen binding. So we get two identical Fabs with our papain digestion. And they also got this tail piece that's made of the rest of the two heavy chains. They named that fraction the FC region. The thing that FC stands for actually is like old-fashioned and no one cares. But if you wanted to have something for it to think about, you could say that it was constant. It is the fraction that was constant. Because we will frequently talk about a constant region, and it happens to be basically this same part. So we'll call it constant region. Yeah? Sure. Fraction or fragment with antigen binding. Yeah. And so you can see the results of the papain digestion here on the right. This is the version from your textbook. You're seeing it from a different textbook on the left. Your fun fact of the day. You probably have not in other classes heard of the protease papain, or thought a lot about the protease papain, but you actually probably know the protease papain. The more common is pineapple. If you've eaten pineapple and you know how you get a tingly feeling in your mouth? It's because it's full of a protease that's actually starting to act in your mouth. And that protease is called papain. It was actually originally isolated from papaya. So that's how it's got that name. And literally the only time I've ever heard of it in my entire life is in a papain digestion of an antibody and in fun facts about pineapples. Like, I honestly don't really hear about it very frequently otherwise. They also did a different kind of protease digestion, and now they used a protease that you might have heard about in other places, which is pepsin. So they also did a pepsin digestion. And when they did their pepsin digestion, they basically got one part of the antibody. And the reason why I say they got one part is because the rest was like totally destroyed. And the one part that they got, you can see, was like the two antigen binding arms connected together. So the degradation happened below this disulfide bond. So basically the whole FC was destroyed. And they got this molecule called a Fab2, that again kind of helped them understand what this overall structure looks like. There are situations where immunologists today will make either Fab fragments or Fab2 fragments or FC fragments to use in experiments. And in fact, I'll mention a little bit more about those a little bit later in the lecture. And so that sort of gave us this understanding of the antibody structure. So antibodies are made of each individual antibody, two identical heavy chains, two identical light chains, that you can see here from your textbook, shown in green and in yellow. You can see your disulfide bonds between the heavy chains, as well as the disulfide bond between the heavy and the light chain. And so that's going to be one of the key parts of this structure that we're going to think about. When you look at these images of the antibody structure, you can also notice that the antibody is drawn as being made of some rectangles. So you can see we've got rectangles depicted here. That actually is immunologist shorthand for a particular type of protein fold or protein domain. And so after people had discovered the antibody and started to really look at the protein, they found out that it has multiple copies of this same protein fold that make up its structure. If you remember on Wednesday, I told you that sometimes we use this term of immune globulin or immunoglobulin when we're talking about antibodies. that fold that makes up each of those rectangles is called the immunoglobulin fold, or the IG fold. And so this is a kind of major protein fold that is found in many of these types of proteins. What you can notice with the immunoglobulin fold is we've got these arrows. So those are all beta sheets that are connected by some loops. So we've got some loops connecting some beta sheets. And we also have a disulfide bond kind of going through the middle. And you can see this in its actual three-dimensional form here, where we've got all these beta sheets kind of coming together like this, and these little loops coming off the top. I was actually taught to think about this as it being like one of those little fancy person sandwiches that has a toothpick in the middle. And so you have two sheets of bread, of beta sheets, and a disulfide bond as the toothpick, holding it together in the middle. So the disulfide bond is kind of like in here, holding this whole thing together, and otherwise it's like a little sandwich made of two sets of beta sheets. And we've got those little loops up at the top. At some different times in the semester, I'm going to mention the loops. So when I'm talking about the loops, different times, those loops, particularly the ones that are labeled there up at the top, are going to be the ones I mean. And you can see this sort of here as well. So this is the FAB of an antibody. So this would be like just this arm. You can see there's like 4 different locations, 4 different little rectangles. So this would be 4 different immunoglobulin folds. And here they are looking like 2 little sandwiches. And you can see there's some loops at the end that are going to be particularly important. This structure of the immunoglobulin fold was first described in the antibody. But now we found that tons of proteins have immunoglobulin folds. And you can see some examples of them here. These are all really important proteins of the immune system. So we see this fold over and over and over and over again in proteins of the immune system. And sometimes people draw it with this little like circular thing with the disulfide bond in between. So if I happen to draw something like that, that might be what I'm trying to draw. I will also say, once upon a time when I was a grad student TAing, I drew an immunoglobulin fold containing protein on the board with little curves like this. I was drawing and kind of not looking like this to look at it, and I realized I had just drawn the most phallic drawing I'd ever drawn in my life, giant on the board. And so I don't tend to draw these proteins as much anymore. So that's kind of one aspect to understand about this antibody structure, is that you can see that there are these different domains that are depicted here as different rectangles. We can also come up with some other ways of talking about different parts of the structure. And one of the biggest things that we often talk about is shown on the right here, which is that the very last immunoglobulin domain of both the heavy chain and the light chain, so my little antibody here, it's the darker colored ones. Here it's the red ones, are actually the place where the antigen binds. That immunoglobulin domain is the really important part. And sometimes we refer to those parts as the variable region. And so you can see they're listed here in red as the variable region. The rest of the light chain and the rest of the heavy chain will be known as the constant region. And so we can also talk about variable region and constant region of our antibodies. And I know this is going to be really shocking, but the variable region and constant region got those names based on how many different, like if I took all the antibodies of your whole body, how many different variable region versions would I find, and how many different constant region versions would I find? I'm going to give you estimates right now. The numbers are totally made up. I will never ask you. In both cases, I'm lying, but they'll give us an ideal. For variable regions, the answer would be about 10 to the 16th. So that is a 10 quintillion. Pretty variable. There's a lot of them. Constant regions, for our purposes, I'm going to say the answer is 5. So not totally constant, but a whole heck of a lot more constant than the variable region. We have names for those 5 different constant regions. So we actually can sort of call those antibodies that have different constant regions different things. The different constant regions, first of all, you can notice from this image and other images, the constant region is not what binds to antigen. The variable region is what binds to antigen. The constant region is what allows the antibody to actually do something. It's what allows the antibody to have its function and deal with a pathogen. And that's going to come up much more later. But when I think about my antibodies and their constant regions, I think of 5 constant regions. Again, there's actually subtypes of some of them, but I'm not really going to worry about those for us. We just got 5. And those 5 are shown here. One of them is called IgM. That means that this sort of the blue domains of the heavy chain are present. There is actually, you will see later, a gene that's involved in IgM that's called the mu constant region. Mu is the Greek letter for M, so they go together. There's also one called IgD, which is encoded by delta. You can see it has these heavy chain constant regions. Notice that when we're talking about this, we're really talking about heavy chains. We're leaving light chains out. There's reasons. That's a later problem. You can see we have one called G, we have one called E, we have one called A. These are, this order of me telling you them is actually an order that matters to us as immunologists. It will not matter to you right now, but later you will want to know MDGEA. And I like to remember this with a mnemonic. You guys have heard the phrase, an apple a day keeps the doctor away. So I learned to remember this with MDs, like doctors. MDs give everyone apples. And that's how I remember MDGEA in terms of the order. For now, you can just use that to remember what the 5 are going forward. When we talk about these antibodies that that differ in their constant region, we refer to them as different isotypes. So this slide, as you can see from the title, is officially telling you the 5 isotypes that exist. And so I might say, what isotype is this antibody? Oh, it's an IgG. Immunoglobulin is the IG. Like, we're just shortening that whole immunoglobulin word and then putting a letter at the end. So those are sort of the options and some of the details I'm going to tell you right at the moment for the constant region. But then I have to tell you some other aspects of some other parts of the structure. So just really briefly, I'm not going to say a ton about this here, but you can notice that there are some additional parts that are not immunoglobulin domains, particularly this region called the hinge region. It's sort of the, shoulders of the Y. And what I want you to just know about the hinge region is that the hinge region is super flexible. In fact, antibodies can wave and change the distance between their fabs. They can rotate their fab arms. I can't really rotate my shoulders super well, but they can rotate this way. They can bend the FC sort of at the elbow. They can even like move the tail. And so we've got lots of flexibility. The antibody is going to be able to sort of reach antigens really well by being really flexible at the hinge region. There's also another piece of information that is really important about when we think about the structure of our sort of stereotypical antibody, which is that we've got two sites that bind antigen, two FAB sites, two variable regions that are identical. So we can actually bind the same antigen twice here. One of the reasons why that is important is that it actually changes some details of what the biochemical interaction between the antigen and the antibody look like. So oftentimes, biochemists think about like on rate and off rate, how quickly two proteins interact and how quickly they come apart, right? We call that affinity. Because the antibody, and so you can see this is kind of what that would look like at the top. Because the antibody has the ability to do this bivalent interaction, to interact more than once, it actually doesn't have a normal on-off rate because you can see both arms can come on. If one arm happens to fall off, it isn't like infinitely able to potentially bind anywhere. It's being held right close to its binding partner and is going to bind on more quickly. So in fact, this has stronger binding than 2 times this. Because you basically have reduced the ability of them to come off, as they are being held really close to other copies of the antigen. And so this is more than double as strong as this because of this repetitive thing. Sometimes we refer to that combined strength as the avidity of antibodies. And so antibodies also have this thing called high avidity because of their bivalent interaction. So the other sort of aspect of the antibody structure that I want to tell you about is some details of the antigen binding sites and how we bind antigen. One of the most important pieces of this is that when we're binding to antigen with our antigen binding sites, that antigen binding site is made of a combination of the heavy and light chain. It's not just the heavy chain doing it alone. It's not just the light chain doing it alone. It's actually completely about the combo of them and sort of the combined interface or the combined shape, the combined structure that they make. And so here in sort of grayish color, I don't know what color that oval is, that's the antigen. So in this, you're the antigen and you're looking at the antibody. And when you're looking at the antibody, you're actually making contact with this part of the heavy chain. So there's an H right there, and this part, as well as you're making contacts biochemically with parts of the light chain. So it's really about the combo of the two, not just which heavy chain is there or which light chain is there. You can see the same thing kind of as a side view here. If we look at the antigen, you can see that the antigen is interacting with both the heavy chain and the light chain. And so what this antibody is specific for is partially based on which heavy chain it has and also partially based on which light chain it has. So you can notice the antigen binds here. Every so often, And I actually understand why this is, but very so often when students try to draw an antibody, they put the antigen here. That's not where the antigen goes. The antigen goes here, making contact with both the heavy chain and the light chain. You can also notice from this diagram that the antibody is actually has sort of 3 different parts of the heavy chain, 3 different parts of the light chain seem to be binding to that antigen. Those 3 different parts are actually those loops I showed you before. The 3 loops from this immunoglobulin domain, the 3 loops of this immunoglobulin domain. So if I go back to my immunoglobulin domain, you can see it up at the top. It's got 3 loops that were holding it together. Those 3 loops on these immunoglobulin domains are actually what are making the physical contact. And so you can see loop 1, loop 2, loop 3, loop 1, loop 2, loop 3 of the heavy chain and the light chain here. Sometimes we even will refer to those loops as CDRs, or complementarity determining regions, because they are the things that tell you which antigen the antibody is complementary to. Yes?
I have a quick question.
Okay.
On the slide you keep pulling up with like the antibody diagram, I think I just kind of confused the colors real quick.
Okay.
So since you said like, because you know when the one on the left is like yellow and green, and then on the right, both of them are red.
Yeah. So what that means is that the light chain has a variable and a constant, and the heavy chain has a variable and a constant. So this is still a heavy chain. This is still a light chain. It's just that in this light chain, there is a variable region and a constant region. The red is variable, the blue is constant. Here, there's a variable region and a longer constant region.
Okay, so like for the one on the left, I don't know why I'm thinking so hard into this.
That's fine.
Like does like the, I guess the orientation, because on that one the light chain is on one side, I guess, on top of each other, and then.
Well, so usually we talk about the light chain as being sort of the outside one, and in a lot of ways that's just the convention of how we always draw it. Is the light chain is always, it's smaller, and we usually draw it on the outside because the heavy chains kind of have to be next to each other to give you this tail. Okay? Yeah. All right. Yeah. Okay.
The 2 constant regions of heavy and light chain, are they like the same?
So I have only mentioned that's a good question. I've only mentioned details about the heavy chain constant region. There is a light chain constant region that for the most part no one cares about. There is legitimately like one spot in one lecture where I'm going to mention it, and there's one place where sometimes immunologists talk about it. So if I am being fully truthful, there are 2 kinds of constant regions of light chains. One is called kappa and one is called lambda. And sometimes you have a kappa and sometimes you have a lambda. And no one cares. So I didn't really mention them, but technically there are specific light chain constant regions, kappa or lambda. Yeah.
Oh, real quick.
Yeah.
So does every light chain and heavy region will have a constant and a variable?
Yes, every light chain has a constant and a variable. Every heavy chain has a constant and a variable. Okay? Yes. I also want to just tell you guys, I have many times in the past told a story of one of the early times, I can't remember exactly which year it was, when I taught at Drew, of a question that somebody asked in my immunology class, which I thought was like the most brilliant question I had ever heard. And I was just like, I can't believe. It was basically like he figured something out multiple lectures in advance. And it was like this question, I was just like, I can't believe he figured that out. And so I've always been like, oh my God, that question. The person who asked it is now a microbiology and immunology professor at TCNJ. Multiple times so far this semester, I have been like, man, these are like Tyler level questions. So like, good job. I've been like super psyched. So I've already basically told you this on that previous slide, but the heavy chain and the light chain actually combine to form the antigen. And so this is just another version of this showing you that really what happens is we have kind of the heavy chain and light chain coming together, making some kind of three-dimensional structure that is complementary to the antigen. When I was a senior in college, I was talking to my roommate who was a political science major, and I told her that actually as an immunologist, all I do is match shapes. It's really like I'm in preschool. Is I ask if shapes match or not. And so you can kind of see that it's the heavy chain and the light chain here. This always shows it as the two of them being supersymmetric, and that's not really true, but whatever, this works. If I were to take all of the 10 to the 16th variable regions from all of your antibodies in your whole body, and I was to do protein sequencing on all of those antibodies or all of those variable regions, I get a lot of differences. Obviously, they're variable regions. But I would find out that the differences were mostly in one part of the variable region. So the variable region isn't sort of equally variable all the way across the immunoglobulin domain. There are parts of it that are extra variable, that are like the most variable. We refer to those as the hypervariable regions, or HVRs. Oh, that's the wrong slide. This is the right slide. And when I actually look at that, you can see I've got 3 areas that are super variable. 3 hypervariable regions. The 3 hypervariable regions are the loops. They actually encode the loop parts. And the 3 hypervariable regions are the 3 little parts that are touching the antigen. So in fact, all the variation really from antibody to antibody is in those loops. And that's how they can each bind to a unique antigen. And so sometimes when I think about an antibody, I think about, on each arm, we've got a heavy chain and a light chain, and they each have 3 little loops coming off like 3 little fingers that are actually the places where they vary and actually change which antigen they bind. And so here you can see kind of those 3 loops shown at the end. Here you can see those 3 loops shown in the structure. Here you can see this in a linear amino acid sequence, that there's 3 parts that vary way more than all the other parts. Yep, Brenna.
So is the CDR loops the
Yes, and so I didn't say that. That's perfect. And so it turns out that the CDR loops, the loops that are the complementarity determining regions, and the hypervariable regions are the same thing. And so those we now know those are actually 2 words for the same loops. So here you can see they're called both CDRs and they're called HVs, or hypervariable regions. So what I hope you might notice from all of this is those loops probably are kind of important. And as we move forward, we're going to care about those loops again. I told you earlier about antibody isotypes, and isotypes being one way that we can divide up the many antibodies that a person has. There are some other terms that we can sometimes use when we're dividing up antibodies. And those are shown on a slide that maybe I can click on. Here. And so sometimes we'll talk about things like idiotype differences or allotype differences. I will say again, this is one of those places where some of you who there are a couple spots when we talk about antibodies where some of you who know a lot of biochemistry are going to look at me and be like, no. Or a lot of us from molecular biology. And the answer is, I know what your problem is, and I promise you I will answer that question eventually. It's just that I haven't totally answered it yet. But I have actually had some students get really confused here. And so what I want you to notice first is that we've got these antibodies from this person named Sherry. I don't know why her name is Sherry, but her name is Sherry. And Sherry could have antibodies of different isotypes, like IgE versus IgG. And so you can see that's the same difference I was showing you before, which is the difference between isotypes. If I was drawing this slide, I would have made this constant region a color that was like different green. Maybe it's darker. I would have made it like purple or something to indicate the difference between IgE and IgG. But they didn't do that. What you can also notice is that we can think about antibodies that have the same constant region, but different variable regions. So here you can see again from Sherry, 2 different antibodies that are both IgG antibodies, but that bind to different antigens. So their constant region is the same, but their variable region is different. And now they did color code this time. And so you can see one of them is an IgG against antigen A, so this might be against influenza, and one of them is IgG against antigen B, this might be against SARS-CoV-2. So same isotype, different variable region gives you this difference in what's called idiotype. So those are antibodies that are different idiotypes. You can also see if I'm looking at 2 antibodies that are basically the same, but are from different people. So here you can see an IgG antibody against SARS-CoV-2 from Sherry and an IgG antibody against SARS-CoV-2 antigen B from John. We refer to those as different allotypes. So 2 different individuals of the same species, but they're same receptor. And the idea here is that like there might be other genetic differences between Sherry and John. Maybe like Sherry has a mutation that changes the amino acid here. And so her antibody isn't actually totally identical to John's because these are just 2 genetically different individuals. And so we would call these antibodies that otherwise the same isotype, same idiotype, but just different from person to person, different in allotype. Yeah, Jonah.
Do those differences cause different, like, functional differences?
Maybe. So there's one other kind of thing related to this I just want to mention. I told you guys last time about the difference between polyclonal and monoclonal antibodies. Some of you had heard the term monoclonal antibodies before. If you remember, the monoclonal antibodies were antibodies that were made specially in the lab, where you have a whole population of antibodies that all bind the same epitope within an antigen. So they're like exactly the same. You had to do some lab purification steps instead of the polyclonal response that you might get. We now use monoclonal antibodies for all sorts of medical uses. We treat lots of diseases by giving people monoclonal antibodies that we've made in the lab. This is a super out of date list of some of the monoclonal antibodies that are currently in use. If you were to actually look at some details of in the pharma industry of kind of classes of drugs and how much money the pharma industry makes off of them and how much the pharma industry plans to like do them in the future, monoclonals win by far. Monoclonals are like where all the money is right now in pharma and what everyone is sort of doing. And so we know a lot about so people in the pharmaceutical industry, even if they're thinking about like heart disease, end up thinking a lot about some of these sort of biochemical details of antibodies because all the hot drugs are monoclonal antibodies. Brenna, did you have a question?
So are monoclonal antibodies allotypes?
So monoclonal antibodies would in fact all be an identical isotype, idiotype, and allotype because we made it in the lab. So it's like a whole bunch of super identical antibodies. And so what you can notice here is we've got some antibodies that are used to treat non-Hodgkin's lymphoma, some breast cancers, some different types of leukemias and lymphomas, neuroblastoma, other types of cancers, arthritis, different types of arthritis, Crohn's disease, ulcerative colitis, psoriasis, multiple sclerosis, asthma. You can see there's like a huge list of diseases where monoclonal antibodies are clinically in use. My guess, I could be totally wrong on this, is that at some point over the weekend, you guys might actually like have the TV on, like see something on TV. And if you do that, you might see an ad for some kind of medicine. Happens relatively frequently. When that happens, I want you to look at the name of the medicine. There are names listed here, and there also will be on the next slide as well. If the name of the drug has MAB at the end, that means it's a monoclonal antibody. So anything that ends in MAB is a monoclonal antibody drug. So yes, for example, you might hear about Humira, and the official name for Humira is adalimumab. So if you see a drug with any of those names, that has MAB at the end, it's a monoclonal antibody. And now that you know that, wherever you might see a drug ad, look for the name of the compound. You will be shocked at how many drug ads you're seeing for monoclonal antibodies that have MABs at the end. We actually also, there's actually some other info in that naming. Because when we think about those antibodies, we have to think about how we make them in a few different ways. So once upon a time, the way that all of our antibodies that were used for therapies were made is they were made in a mouse. You were like, okay, mouse, I need you to make an antibody against this protein, and I'm going to use it for therapy. And so we got an antibody that came out of a mouse. And then we injected that antibody that came out of a mouse into people to treat them. And in many cases, these drugs had like crazy positive effects, like life-changing effects. But sometimes when we did that, they also had some side effects, because you were pumping a person full of a mouse protein, a foreign protein. At some point, your immune system was like, what is this mouse protein doing that you put in? And so we eventually figured out ways to try to make the molecule more and more of a human antibody and not a mouse antibody. So we changed first to have just a human constant region with the mouse variable regions. Or to have the human everything but the CDRs and just the mouse CDRs. Or eventually to have the entire thing be a human antibody molecule. It's kind of tricky. You might imagine that I probably, if I want to make a humanized antibody for a drug, say I want to make grams of an antibody against SARS-CoV-2, I'm not going to inject you full of tons of SARS-CoV-2 and collect your antibodies. I have to do a lot of lab work. I'm not going to have a human patient who's making these antibodies for me. And so there's sort of a lot of biochemical work that's been going on here. But actually, if you look at the name of the antibody, the MAB, the part that comes before that tells you what kind of antibody it is. If it's a mouse antibody, if it's a chimeric, if it's partially humanized with just the mouse CD3s, or if it's a fully human antibody. And so you can see UMAB is a fully human antibody. ZUMAB is humanized. You can see, usually it's, IMAB is the chimeric, and OMAB is the mouse. And so, again, this is one of those things where you will start to see some of these, these drug names. Now that you know it's a thing, if you see a drug ad, look for that. And you'll be surprised at actually how much information is given in that drug name, and how many monoclonal antibodies there are being used. One of the ways that we're actually able to do fully humanized antibodies now is someone actually generated a mouse that has the entire human antibody genetics section. So we basically put the entire human antibody gene region, which is a massive region of the human genome, into mice, so that we can have mice make human antibodies for us. And so you can see these mice are known as the velocimune mice. And this was 10 years ago. There were already 10 therapeutics with the velocimune mice, and they were probably the most valuable mice ever engineered, having already brought in $2 billion at that point. And they just keep on going. There are other situations where people have done things like say, you know what? Using an antibody for therapy, this protein that we're trying to use as a therapy for somebody is actually too big of a protein. This protein actually can't fit in the special anatomical location where we need to do treatment. We need something even smaller than this protein. And what they've ended up doing is they've ended up designing things like just Fabs or even just variable regions in the lab and injecting people with those. Some of this, we figured out how to, and sometimes we even do heavy chains alone without light chains. It turns out that there are some organisms that are super weird that only have heavy chains and don't have light chains. The most famous organism that only has a heavy chain and no light chains, and it's antibodies. Can I tell you why? No. But it's a camel. Camels only have heavy chains. A camel biologist. I do not know who found that out. And so based on looking at the structures of, say, those camel antibodies or otherwise trying to make like just FABs or just FCs, there are even times where people will make FAB2s so you can get like cross binding, but you don't have the tail in the way, will be used for a lot of different therapeutics. Officially, these are all called nanobodies, which are like these further modifications of antibodies based on all these different structural parts. So at this point, we've seen the structure of an antibody, but we haven't totally talked about the function of an antibody. And if you read online stuff, especially kind of during COVID and things, you would not believe the verbs people use with antibodies that they think antibodies can do. Antibodies sound like freaking superheroes in a lot of cases. Like they're wearing a little cape and like they're doing all sorts of crazy things. But really, an antibody is a protein with 4 chains. A protein cannot eat things, right? There are kind of limits to what you can actually do to get rid of a microbe if you're just a protein made of 4 chains. And so what we have to think about is how in the world does this little protein actually help us get rid of a microbe? There are a bunch of answers to that question, which are known as the antibody effector functions. I always think of them as the 5 antibody effector functions. This image from your textbook, or from a different textbook, says there's a 6th one. I kind of get where they're going with the 6th one, so we can have 6, sure. Let's have 6. Different antibody effector functions. And so we need to see these 6 different antibody effector functions. The first one that I'm going to tell you about is pretty easy. And the reason why I'm telling you about it first is because you actually already know it. You just don't know you know it. So one of the ways that antibodies can deal with a pathogen, one of the effector functions of antibodies, is that antibodies can start the complement cascade. So one of the reasons why antibodies can be actually useful in getting rid of a microbe is that they can start the classical pathway of complement, like we already learned about. So fixing complement or starting the classical complement cascade is one of the ways antibodies can get rid of stuff. Yay. There is a second one that you also kind of already know about. And that second one is opsonization. So you have heard of opsonization when we talked about complement as well. At that point, you heard about how complement can do this thing called opsonization. And when I talked about that, I said that opsonization is when you coat something with something else and make it more likely to get phagocytosed. I told you it was like butter. When you put a coating on something to make it look more tasty and more likely to get eaten. So you could opsonize or coat a microbe with complement, and that would make it more likely to get phagocytosed and more likely to get eaten. It turns out if you coat a microbe with antibodies, it does the same thing. It also opsonizes. It also is a coating on that microbe that makes that microbe more likely to get eaten. So it's a different kind of butter. And so the second way that antibodies can deal with microbes is that they can opsonize those microbes. When we talked about opsonization and complement, we specifically, I had to tell you that there are some receptors on the surface of some phagocytic cells that were complement receptors. And that was how the phagocytic cell knew there was complement on the pathogen surface and knew to do phagocytosis because there was a complement receptor. Well, now our coding is an antibody, so complement receptors may not be super helpful. So what you might notice is there probably should be an antibody receptor as well. There is an antibody receptor. There are multiple types of antibody receptors, in fact. And all of those receptors for antibodies bind to the Fc portion of the antibody, which we can again, remember I told you shorthand-wise it's constant. It's this tail part down here. These receptors that are binding to the Fc portion of the antibody are called Fc receptors. And so we can have opsonization happen because phagocytic cells have Fc receptors on their surface. You can see a bunch of different Fc receptors here. Many of them are immunoglobulin domain containing proteins. And you can also notice that some of their names have things like Fc epsilon, Fc alpha, Fc gamma. Fc epsilon binds IgE, the E one, because E and epsilon, E is the Greek letter for epsilon, or epsilon and E are the same, and I don't know how I phrased that a second ago. Similarly, the Fc alpha receptor binds IgA. The Fc gamma receptor binds IgG, and so on. So that's 2 of our 6 functions. The third of our 6 functions is also one that I find relatively straightforward, which is called neutralization. We often think about neutralization as being a really useful function when we are thinking about viruses. And so neutralization comes up a lot when we're thinking about viruses. I, as an immunologist who thinks about virus infection, think about neutralization pretty often. A lot of times, virologists will look at their antibodies and they will do some experiments to see if their antibodies can do the function of neutralization. And if they can't, they're like, these are trash. And we're learning more and more, oh, right, the other ones matter for viruses too. It's not just neutralization. Maybe we shouldn't make the other ones be trash. There are those other 5 functions after all. Oops. This is a debate that's happening sometimes in the field. But to understand neutralization, we can look at this slide that's shown here. And On the left, we're seeing a situation where there is no antibody and there's no neutralization. And what you can see happens is we've got this virus. It's going to bind to something on the surface of the cell. That's going to let the virus into the cell to infect and cause, terrible destruction. If, however, there are antibodies around, those antibodies can bind to the virus and prevent that virus from being able to interact with our cells. Basically, this antibody is blocking the virus from being able to interact with the receptor on our cell. And so this virus can never get into cell and never cause problems. So neutralization is this physical blocking, the antibody physically blocking, in this case a virus, from interacting with a receptor. That's what neutralization is. Sometimes I just have described neutralization in my life as a fancy word for steric hindrance. Because it's really just this antibody is getting in the way of these other two things binding. And you can see this is a sort of similar figure. This is actually a structural reconstruction of rhinovirus, which is one of the things that causes the common cold. And here in green, you can actually see antibodies that are binding to that rhinovirus. And if this little spot is what needs to interact with the receptor, if you just put an antibody in the way, then we can't have receptor binding. We can't have viral replication. We don't get a cold. Hooray. And that binding can be to the exact same spot, or it actually can just be close enough that it structurally is in the way. One other place where neutralization is really important is in the case where we have toxins. So some bacteria make toxins that cause us a lot of harm. Some other situations involve toxins that can cause us a lot of harm. And so that toxin can interact with some receptor on the surface of our cell and then can cause us harm. We can have an antibody that can neutralize that toxin so that the toxin cannot interact with its receptor and not cause us any harm. And so neutralization is a great way that antibodies are able to protect us against some of these microbes. Yes?
When the antibody neutralizes anything, does it just float around with it on it, or does another cell come and kill both of them?
Great question. So they're generally not going to be necessarily alive, so it's hard to say that they would get killed, but generally they're going to get degraded. So they're going to float around for a while, and they're probably eventually going to get cleaned up as random trash. Yes?
I was wondering is there a specific area on the antibody where there are usually more receptors?
So the antibody just has the two FABs, and their job is just to bind antigen, and then there's the FC. And so usually what we think about in a case like this is that the FC is in the way. Is really what the idea is. It's just that the FC is in the way of this other kind of binding. Okay? So think of the FC as sort of this bulky, annoying thing in the way. So that's 3 of our antibody functions. This is the 4th one. No one likes it. It's really lame. The basic idea here is that if you have many antigen molecules and many antibody molecules, because oftentimes those antigens will have repetitive epitopes and the antibody has multiple arms, you can sometimes get these giant clumps of multiple antigens that are bound to multiple antibodies. And so you can see a nice example here. These are known as antigen-antibody complexes, these clumps. And when those are made either in blood or in tissues, they basically precipitate out. They basically become a whole bunch more trash to get rid of. And so it's sort of like we're taking this antigen and we're pulling it out of the blood. We're pulling it out of the solution by putting it in this big trash pile of making this antigen antibody complex. And so, sort of as was mentioned before, usually what happens if you have this big trash antigen antibody complex, it usually gets cleared by phagocytosis as trash. So that's the fourth one.
No benefit?
No, what do you mean no benefit?
Like it's just, they're just clumping up and then
It's basically just getting rid of antigen is the only benefit of it. Then there are two other ones that to mention to you. One, and they both have something in common. In fact, they have this thing in common with opsonization. And this is that this, as well as the one I'm going to show you on the next slide, also requires an Fc receptor. So you also have to have a cell, in both cases, that has a receptor on it that's binding to the Fc portion of the antibody. So the antibody is kind of signaling to some cell with an Fc receptor. Here, the cell with an Fc receptor is called an NK cell, or a natural killer cell. If we have antibodies bound to something and we bring in an NK cell, that NK cell, as you might guess from its name of natural killer, will kill. And so basically the antibody is sort of tagging whatever as a thing that it wants the NK cell to come kill. Sometimes we refer to this as antibody-dependent cellular cytotoxicity, or ADCC. It's also, if you were going to write a definition of ADCC, you would say it's activating NK cells. And the final function is really the same thing. It's just that we aren't going to have an NK cell at this point. It's now we're going to be seeing a mast cell get activated. So we can also have mast cells that have Fc receptors on their surface, and if they're binding to antibody and to a microbe, that mast cell can be activated. And so you can see the title of this one, it says activation of mast cells, and the definition of activation of mast cells is activation of mast cells. So it's really tricky. As you will see on Monday, each of these different isotypes has different kind of uses. Each one is like really good at some function and kind of meh at other functions. So there are different times when you might want different isotypes.